Good morning, students. Now coming to co-dominance. Students, in normal monohybrid cross, F1 hybrid it resemble with either of the parents, only one of the parents phenotypically. That is, dominant phenotype is established and expressed in F1 generation. But in case of incomplete dominance, what happened in incomplete dominance? F1 hybrid it, did not resemble any one of the parents. It was between the parental phenotypes, means third phenotype which was not present in which is not the parental phenotype. It is expressed in F1 hybrids. But in co-dominance, okay, in co-dominance, F1 hybrid it resemble both parental phenotypes. Both parental phenotypes are established in F1 generation. Expression of both parental phenotypes in F1 generation, it is considered as co-dominance. Both parental phenotypes they are established, they are expressed in F1 generation. That is considered as co-dominance. Students, this in this type, both parental phenotypes, they expressed in F1 generation. Both parental phenotypes expressed in F1 generation. This is considered as co-dominance. Expression of both parental phenotypes in F1 happen generation is considered as co-dominance. One of the best example for this co-dominance that is the different kinds of red blood cells of humans. Different kinds of red blood cells in human beings that determine ABO grouping, blood grouping. Okay, that determine ABO blood grouping. They are best examples for these co-dominants. When it comes to the plants, there are some examples in plants also. For example, seed pod pattern. Okay, seed pod pattern of plant, seed pod pattern of plant, seed pod pattern and size in lentils, size in lentils. They are the best examples. Okay, these are the best example for lentils. These are the best examples for this co-dominance. Okay, so seed pod pattern and the size of lentils. Example, this is lens culinaris. Okay, lens culinaris and sub species like culinaris. Okay, culinaris is also called medic and also Williams. They are the best examples for these co-dominants. Anyhow, some of the best examples for these co-dominants. ABO blood mean different kinds of red blood cells in human beings that determine ABO blood grouping. At the same time, the seed pod pattern and the size of lentils, lens culinaris subspecies like culinaris and Williams, they are the best examples. Mendel conducted to explain this co-dominance in this lentils. Mendel conducted one homozygous spotted lentil. Okay, one homozygous spotted lentil. Okay, one homozygous spotted lentil. Homozygous spotted. This homozygous spotted lentil, it is represented by large irregular shaped patches on seed pod. Large irregular shaped patches on seed coat. This is a homozygous spotted plant with a large irregular shaped patches and seed coats crossed with another homozygous dotted lentil represented by very small dots on the seed coat like this. Okay, now one homozygous spotted plant is crossed, one homozygous spotted plant with a large irregular shaped patches on seed coats crossed with another homozygous dotted plant having the small circular dots in the seed coat. Now these two plants are crossed one to each other. At the same time, the genes responsible for this spotted character, such genes are considered as CS and CS genes are responsible for spotted character. Similarly, dotted characters alleles are CD and CD. They are alleles. So these are parents. Okay, and these are the alleles. Okay, so spotted character alleles are CS and CS, and dotted character alleles are CD and CD. Now these two plants are crossed one to each other. And what type of gametes are formed from these two types of parental plants? Here CS gametes are formed. Okay, CS gametes. And here CD gametes are formed from the second parent plant. So two gametes CS gametes are formed, and also CD gametes are formed. As the combination of these CS it leads to CS and CD, and this it leads to the second gamete. This is CS and CD. These are considered as F1 hybrids. F1 hybrids. Okay, these are considered as F1 hybrids. So the alleles present in F1 hybrids CS CD. And what is the phenotype of this F1 hybrid? Normally monohybrid crosses dominant characters are expressed in F1 generation. For example, in case of incomplete dominance, F1 hybrid did not resemble any of the parents, but here the parental characters are expressed in this type. For example, this is the seed. This seed coat is also represented by large irregular patches, like the first parent. At the same time, this is also represented by small circular dots like second parent. So this is considered as spotted and dotted alleles are there. Spotted and dotted characters they are expressed similarly. They are expressed equally in F1 hybrids. So these type of incomplete in codominance, both parental types they are equally expressed in F1 generation. Expression of both parental phenotypes in F1 generation. This is considered as co-dominance. Spotted character and dotted character both are expressed in F1 hybrids. Okay, students, while both spotted and dotted characters both are expressed in F1 generation in this co-dominance, here neither spotted alleles nor dotted alleles are recessive and dominant over the next. Means neither spotted in this codominance, neither spotted nor dotted alleles are dominant and recessive. Dominant or recessive over the next. Spotted character is dominates over the dotted character. Irreversibly, dotted characters is also dominance over the spotted character. Both characters are neither spotted nor dotted alleles are dominant or recessive over the next two. Both characters are equally dominated to each other. Because of this reason, both characters are established in a F1 generation. Expression of this both parental phenotypes in this F1 generation is considered as codominance. Mendel also selfed these F1 hybrids, and also Mendel selfed F1 hybrids and also obtained F2 progeny and also explained the phenotypes and genotypes in F2 progeny. So here this is CS and CD considered as male, and this is CS CD considered as female. Now these F1 hybrid produce two different kinds of gametes. One is CS gamete and second one is CD. And this plant also produce two types of gametes. One is CS and other one is CD. They are the gametes. So here selfing. This is selfing of F1 hybrids. These plants produce two different kinds of gametes with the CS and CD, and it also produce another two gametes of CS plus CD. So they are the gametes. Now combine them and find out the genotypes and phenotypes. Okay, here these CS may be combined with CS may be combined with CS, leading to the formation of CS. Now this gamete also has 50% of chances to fertilize with CD. Now this gamete has 50% of combinations to form CS-CD, and is also contain 50% of chances to fertilize with CD. In this way, total 4 plants are formed in F2 progeny. Now this F2, this is F2 generation. This is F2 generation. Now the combination of these gametes and formation of a progeny and their phenotypes and genotypes can be explained by using Punnett square board like this. This is a Punnett square board that explains the combination of these gametes. Punnett square board. Okay, this is a Punnett square board that explains the combination of these gametes. These male gametes written on the top row CS and CD. Similarly, female gametes can be taken in a left row CS gamete and CD gamete. Now combine these gametes and find out the genotypes and phenotypes and what is the phenotypic ratio and genotypic ratio of these codominants. Now CS, okay, then CS, CD, then CS, CD, then the next combination CD. Total 4 plants are formed as a result of the combination of this gamete set in a F2 progeny. And find out the phenotypes. They are the genotypes of this F2 progeny. But what are the phenotypes? CS. This is similar to the first parental plant. This is a homozygous spotted lentil. Homozygous spotted lentil. And what about the CS-CD? CS-CD, it is resemble with the F1 hybrid. Heterozygous. This is a heterozygous. And this heterozygous, it is represented by both spotted and dotted characters. Spotted and dotted characters. But what about the fourth plant? This fourth plant is similar with the CD, homozygous dotted. This is homozygous dotted alleles. In this way, total 4 glands are formed. One is homozygous spotted lentil. Two are heterozygous spotted and dotted. And one plant is dotted, purely dotted, homozygous dotted plants. In this way, 25% of the plants are spotted and 50% of the plants are spotted dotted, and only 25% of the plants are their dotted characters. Now, the phenotypic ratio, okay, phenotypic ratio. How many phenotypes are there? There are total 3 phenotypes. One is spotted phenotype 1, okay, and 2 plants are spotted dotted alleles, and only one is dotted. Phenotypic ratio in codominance 1:2:1. But what about the genotypic ratio? Genotypically, these plants also 3 types. Genotypic ratio. Here, one is CSCS homozygous alleles. 1. 2 plants are CSCD heterozygous in condition, and only one plant is CDCD homozygous in condition. So, phenotypically, genotypically, ratio is the same in codominance. Phenotypic and genotypic ratio in codominance 1:2:1. Phenotypic and genotypic ratio 1:2:1. This is the codominance. So, phenotypically and genotypically ratio is the similar. Very important, students. You know the phenotypic and genotypic ratio of incomplete dominance. What is the phenotypic and genotypic ratio of incomplete dominance? Phenotypically and genotypically ratio is also the same in incomplete dominance. That is 1:2:1. So, phenotypic and genotypic ratios of not only incomplete dominance but also codominance, they are exactly the similar. That is 1:2:1. This is codominance. Students, now coming to another point, very important. That is, according to the concept of Mendel, every character is controlled by one gene. Means, one gene is responsible for the expression of one character. But sometimes single gene may be express multiple characters, that is more than one characters. Single gene is responsible for more than one characters. That is considered as pleiotropy. This is called pleiotropy. This is pleiotropy. What is pleiotropy? Only one gene. Sometimes it may be responsible for the expression of multiple characters, that is more than one characters. That is considered as pleiotropy. Some of the examples, one of the best example for this pleiotropy, that is the synthesis of starch. Okay, the synthesis of starch. Synthesis of starch in pea plants. This is under the controlling of one gene having 2 alleles. One is capital P and other one is small p. So, synthesis of starch in pea plants, synthesis of starch in pea seeds is under the controlling of one gene having 2 alleles, that is capital B and small p. When these alleles are present in a homozygous condition, that is, when these alleles are present in a homozygous dominant in a condition, it is responsible for this dominant capital B, they are responsible for the expression of 3 characters. One is efficiency, okay, efficiency of starch synthesis. Okay, efficiency of starch synthesis, high efficiency of starch synthesis, and is also responsible for the seed shape. This is a round shape. Round shape. Okay, shape is round, and it is also responsible for the size of the seeds. Large sized seeds. Large size. Okay, these when the genes are present in a homozygous dominant. These homozygous dominant alleles capital B, they express three characters. One is highly efficient in starch synthesis, and round shaped seeds, and large sized seeds. But if these alleles are present in a recessive condition, that is in a small b, is also responsible for three characters: less efficient in synthesis of starch. Okay, less efficient in starch synthesis. At the same time, these are wrinkled seeds, and size is very small sized seeds. Very small sized seeds. This is the characters. They are expressed by the recessive alleles. So, gene is the same gene, one gene, having two alleles capital B, small b. But here single gene is responsible for multiple characters, that is more than one characters. When the genes are present in capital B, they show high efficiency starch synthesis, round shaped seeds, and large sized seeds. If they are present in recessive condition small b, less efficient in starch synthesis, and wrinkled seeds, and small sized seeds. Here, very important, when these two plants are crossed, when obtained the capital B, small b hybrid, F1 hybrid. Here, what characters are expressed in F1 hybrid? Normally in monohybrid crosses, dominant characters are established in F1 hybrid. But what are dominant characters? Highly efficient starch synthesis. Here also, high efficiency in starch synthesis tubes are found. Then, here, round shaped seeds, round shape is dominant character. Also established in round shaped seeds. But this dominant character large, it is not expressed in F1 generation. And small character is not expressed in F1 generation. So medium sized seeds. Okay, medium sized seeds. Medium sized seeds are established in F1 generation. So only two characters of these three characters. Only two dominant characters are established in F1 generation, but not the third character. That is the size of the seed. Intermediate size, medium sized seeds are formed in F1 generation. Means here, when the size of the seed is considered as the dominant character, this is one of the example for incomplete dominance. Why this is considered as incomplete dominance? If the size of the seed is considered as the dominant character, large size and small size parental phenotypes, they are not expressed in F1 generation. Here, medium size, which did not resemble with their parents, medium sized seeds are found in F1 generation. If in case of the seed size is considered as the phenotype, this is one of the best example for incomplete dominance. When the size of the seed is considered as the dominant character, this is one of the example for incomplete dominance. Incomplete dominance. So, definition is very very important, students. What is pleiotropy and what is example for this pleiotropy? Sometimes a single gene, occasionally single gene is responsible for more than one characters, multiple characters. That is called pleiotropy. One of the best example for this pleiotropy, the genes responsible for the synthesis of starch in pea seeds. Such a example, it has one gene having two alleles, capital B and capital B. Okay.